Hey guys, hi, this is me, Sona saying welcome to GeeklyEDU, and today we are going to talk about cell cycle and cell division, and particularly there are two types, just in case you're wondering. And this particular video will deal with the events of cell cycle, its regulation, what is cell division, and particularly we'll be talking about mitosis. There are two types, mitosis and meiosis. Meiosis would be dealt in another video, and you'll also get homework problems for mitosis, right? So let's get started. What are we waiting for? So what is cell cycle? So you have events in your life. Similarly, a cell also has events in the life, right? So cell cycle is the summary of the events that happen in the life of a cell. So let's talk about one cell that's zygote. It grows and ultimately divides to form two cells, and these keeps on going and going and going, right? And the products when the cell divides is called daughter cells, right? And daughter because these new daughter cells can divide again, but again some cells want to take a stop. Sometimes they don't want to divide further, they rather differentiate, pick up a special job. Like for example, you stop studying all subjects and you just focus on one subject. That's a special job. Let's say some dividing cells become a red blood cell, which is nothing, just a bag of hemoglobin, so all other organelles are discarded, and this hemoglobin carries oxygen. So cell cycle surmises all of such events, which may or may not include always a division. So if this chart represents the cell cycle, a part of it is the division, right? This is called the M phase, or simply the dividing phase of the cell. Well, when scientists started studying this kind of structures, they thought that under the remaining part of the cell, it does nothing. So under the microscope, they could see the movements of chromosomes during the M phase. Now, what is chromosome? That's protein and DNA. And if you've forgotten, don't worry. There's a separate lecture on that. I'll leave the link in description. So cell appeared to be busy moving these chromosomes, and the remaining part cell appeared to do almost nothing because you cannot observe chemical reactions, right? So scientists call this interphase, although this is wrong. The cell is in fact very busy during this interphase, and we will study about that. So this interphase can be further divided into more stages. The first one we want to call G1, then S, then G2. G1 stands for nothing but gap 1. So what exactly happens in gap 1 is that the cell is growing, it's increasing in size, doing its job, perhaps making proteins for its work, whatever is its job. For example, if we want to talk about RBC, it makes hemoglobin, right? So whatever proteins or whatever structures it needs to make or do, that is what it's doing. And then there's a checkpoint. Checkpoints you'll find are basically the decision boundaries where cells decide, do I want to divide or do I want to stay here? So it might be a yes or no question for the cell. So what happens when it says no? So let's take an example. So hematopoietic stem cells, they are found in your bone marrow. They keep on dividing to produce new RBCs, right? So certain times these cells will divide, and there will be two daughter cells. One will say yes to the checkpoint, another will say no. It will stop. It will specialize. It will pick up the task of carrying oxygen by producing just enough of hemoglobin, right? So that is what is differentiation occurring here, and this is called G naught. Now G naught may sometimes be terminal, so we say terminal differentiation has occurred. That means once this RBC is formed, there's no going back. Certain times some cells can just arrest themselves, so a temporary arrest can be there, and this is not terminal, right? Sometimes the cell can just take a brief pause into G naught and can then again come back, say yes, and then go on to the S phase, which is the phase where the cell duplicates the DNA, the code. It has to be copied, right? Because DNA has all the information, right? So S phase is the phase where the cell duplicates all its DNA so that it can finally distribute it to the daughter cells. And don't worry, we'll get to the details. But I want you to remember that as the result of S phase, now you have two chromatids per chromosome, right? Next comes the gap 2. This is where the cellular machinery is being prepared. So the duplication and the preparation for the cellular machinery starts, right? For example, at the end of S phase, you'll have the centrosomes duplicating and other stuff, right? And finally, you again have the M phase. Now, The M phase or the division can be carried as two forms. There's mitosis and there's meiosis, right? This particular video will be dealing with mitosis, but the differences are like mitosis gives you two daughter cells, each with the same number of chromosomes. Meiosis gives you four. So mitosis maintains the chromosome number. If it's 2, that means it's 2. Whereas meiosis reduces the chromosome number to half. The mechanism we'll see another video, right? But you have to ask why. Why? What's the significance? So let's assume there's a father and there's a mother, right? And we already know that fathers produce sperm, mothers produce ovum. Now let's say that these sperms and ovum are produced via mitosis, and these are to unite to form a zygote. So 46 is the sperm, 46 is the ovum, zygote becomes 92. Zygote develops into the daughter. Daughter again goes for a marriage with another guy who is just second generation, just like she is. He's 92 as well. Baby ends with 184 chromosomes, and this keeps on going and going and going. Where will you fit all these chromosomes, right? So this is just impractical. So that is why we have meiosis. So sexual reproduction always happens with meiosis. So meiosis reduces the chromosome number to half. So sperm and ovum ends up with just 23, and zygote with 46. And at each step, the gametes which unite to form a zygote will end up with half the chromosomes. So you see, meiosis is necessary, absolutely necessary for sexual reproduction to happen. That is the importance of meiosis. Now let's jump back to mitosis once again and talk about it in as much detail as possible. So when you talk about mitosis or even in meiosis, you have two stages, karyokinesis and cytokinesis. I would like to remind you, kinesis means you're breaking down. Carry on comes from kernel, that means the core, that means the nucleus. So mitosis is the division of the nucleus, whereas the cytokinesis is once the nucleus, that means the chromosomes have divided, you are going to separate the cytoplasm. Right? So karyokinesis implies the separating out of the chromatids, and I want you to remember that chromatids are the Xerox copies, right? They are the duplicate copies, okay? And once you're done with that, you're going to divide the cytoplasm, right? Now, the chromatid production happens in S phase, remember, and there's another video on how you just copy the DNA. That's called DNA replication, and I'll leave the link below in the description. Don't worry about it. Okay, so let's start talking about karyokinesis. That happens via 4 phases or stages, you can call them: prophase, metaphase, anaphase, and telophase. And we got to understand each step one by one, and then we can go to talk about the regulation of the cell cycle. So let's assume this is a cell, and it has these little centrosomes. Remember, the centrosomes started duplicating at around the S phase. So there's 2 now, right? They are centrioles which are almost perpendicular, and by this time they have started giving out microtubules. These microtubules are called astral microtubules. They look like rays of sun coming out of them, right? Now they have started moving towards the poles. Here I have already showed them at poles, but remember, at this time they have just started moving towards the poles, and then there is nucleus which has almost started to degrade, hence the gaps. So the nuclear membrane will have started to disintegrate, because we need to bring all the chromosomes into the center so that we can pull apart the chromatids, right? So humans, let's say we have 46 chromosomes. We do have 46 chromosomes, 23 from mom, 23 from dad. So this is what it looks like at interphase. It's called chromatin, that is DNA associated with proteins, right? The diameter is hardly 300 nanometers. That's what it will look like at interphase. You can't actually see them. It's so fine, right? Together with the histone proteins. But the condensation starts in the prophase. That means these chromatin material, they form, they coil around, they form loops, super loops, right? You can see those forming loops. And ultimately, this looping, this condensation would bring them to the stage of what you call as chromosomes, when they would start becoming visible. So if we have 2 chromatin, they will wind up to have 2 chromosomes. But what is peculiar is all the chromosomes are going to have two chromatids attached to it, and this assembly is around 300 nanometers, right? So let's say the first one was the original, the second one is the copy, a duplicate, which we produce by replication. And these are called sister chromatids. Originally, they are not sisters because sisters will have differences, but these are exact replicas, right? So by the prophase, you can see the chromosomes. By the end of prophase, you'll be able to see the chromosomes because they have super condensed now. Not all the way down, but they have condensed enough that it's visible under the chromosome, under the microscope, right? So that is the prophase of this, right? The centrosomes, they are not found in all cells. Remember this. So here I have shown them, but like plant cells, they don't have. So they have something called microtubule organizing centers. So what happens is, like, these microtubules, they have a negative and a positive end, right? So what this negative and positive simply means is the positive end is the one where you add microtubules, and the negative is where you add a tubulin units, and the negative is where they break off, right? So these negative ends, they are sort of accumulate towards one end, and this is aided by motor proteins, and these motor proteins sort of hold them together, right? This is one of the ways, but there are other ways. Some evidence even finds them that they might be even linked with some some chromosomes helping them do this, right? So we have a lot of new research coming up. Now, what happens to Golgi and ER? So they disintegrate. This is what we used to believe. Some even say that some of them, they merge, like the Golgi merges with the ER for temporary time, or they even divide and distribute themselves. So different opinions or theories are emerging now, but the one that you read in books is like they simply disintegrate and they are formed. So this was all about prophase, and now we must jump to the next one, that's metaphase. So the job is to bring all of these to the center, right? All of these to the center. Why? Because once they are at the center, it's easy to pull them off, right? So there is a constriction you can quickly see here that's called centromere. Its primary constriction is the first constriction that you can see. And why exactly it's constricted? Because it has repeated sequences, right? And these sequences where the chromatin material is closest, and just outside that, on both the sides, you have a protein assembly that we call kinetochores. Now these kinetochores are very important because this is where you are going to attach the microtubules. So microtubules are like tethers, right? So they are going to pull apart the chromatids eventually, right? So you got to remember these are kinetochores. Now, how all of this is brought to the center? So the microtubules, they are organized and they sort of, there's a tug of war that brings them to the center. There's a constant push and pull going in the directions that sort of tends to bring them closer. Now you can see that it needs not be a single thread, right? It can be multiple threads together linking them. Okay, so ultimately, some of the threads are going to shorten and some of them are going to elongate. The threads, the microtubules which are linked with the chromosomes, we call them chromosomal microtubules or kinetochore microtubules. Just the reason because they're directly linked with the chromosome, and they might be first attached to the sites before directly being attached to the kinetochores, right? And the other ones that join the opposite centrosomes, they are called the polar microtubules because they join the polar. Okay, so the job is that one end, the relative shortening and lengthening of these microtubules will try to bring them to the center. So if if a particular chromosome is more towards the left, so the left microtubule is going to elongate more, and the right one is going to shorten more, right? So they want to bring it back to the center. That's called equator. And how we do this? By relative degradation and polymerization of the microtubules. So the polymerization degradation, there is a balance, and the balance fluctuates in the direction where we want to bring them close, or where do we want to shift them. So that is how they operate. Now, so let's say one ends, they polymerize slower, and the other ends they degrade faster, right? So in that position, the net movement will be towards the left side. Okay, so this is relative degradation and polymerization of microtubules. Okay, now remember, as a result of all of this, we want to bring them to the center so that finally now we can pull them apart and our job is done. So this is what happens by the end of metaphase, and this is when we want to when the chromosomes are the best visible, we want to click the micrograph right now, and we want to take a picture that we want to call karyotype, where we arrange the chromosomes from the largest to the smallest, and we also want to identify if there's any chromosomal disorders that that is there in the cell at this point of time, and is widely used in genetic counseling. Now, the third phase is called the Anaphase. Anaphase is when the chromatids finally separate off and are directed towards the opposite ends of the pole, right? So, again, the plus ends, they start to lose tubulin units as well, and this is a key difference from metaphase, and this is why they are finally pulled, right? So chromosomal microtubules, they shorten up. The polar microtubules, they allow expansion, so it's like they stabilize the whole thing. They maintain the whole thing, and the chromosomal microtubules, they sort of try to pull apart, right? Okay, so the polar ones, they stabilize, and the chromosomal ones, they pull them apart, and there's proteins, there's cross-links that stabilizes the whole spindle. Now, often you'll find anaphase A and B. A is when the centromeres are pulled apart and the chromosomal microtubules shorten, pulling the chromatids to the opposite directions. Whereas in the anaphase B, the empty OCs start moving away. That means the whole polar microtubules, they'll start sliding away from each other, right? And why all of this happens? Because of certain proteins that can literally walk on these protein threads. Now that they have split apart, the chromatids have been split apart, we can totally call them chromosomes. Now you must understand that before, even though they were two chromatids, they were attached to each other at the point of centromere. So that is why we were calling them just 46 chromosomes. They were 46, so they were still 46. But now you can call them chromosomes. So for that brief period of time during the anaphase, you have double the amount of chromosomes in each cell until the cytokinesis part takes place. Now, before that, there's telophase. Now, several times telophase and cytokinesis overlap. So, nuclear envelope starts to reform, chromosomes decondense, and then it's back to at the poles, right? So, many times they overlap. Now, let's talk about cytokinesis. Cytokinesis in plants and animals is slightly different, because you can imagine that in animals it happens via furrow formation, whereas in plants a cell plate develops, and this is the primary difference. Now, why the furrow is forming? That is because of a contractile ring of actin filaments and several bipolar myosin proteins that sort of pull the plasma membranes closer. Whereas in the plants, a cell plate called phragmoplast forms, and that can start as an early depository plate upon which the cellulose and the hemicellulose and all of that gets laid and a cell wall is formed, right? So this is a primary difference between animals and plants undergoing cytokinesis. And once the cytokinesis is done, you have two daughter cells, each with the same number of chromosomes. Now, there's a very important part of control. You can't just have your cells dividing anytime they want. So there's cyclin and cyclin-dependent kinases. Remember, kinases, they transfer phosphate group, right? So these cyclin-dependent kinases, they become active when cyclin is attached. And what they do is they activate something, they modify something, and they modify the action of a protein, and this is how they control the cell cycle. So G1S and G2, this is when the cyclin concentration keeps on increasing and it passes a certain threshold, and that is when the M phase kicks in. Now, we have several checkpoints. As you can see, the G2M transition is checked. It's influenced by cell size, DNA damage, DNA replication. But remember, these are gross events, right? So any of these can alter one or the other transcriptional changes, right? So several proteins would be released and which would change the cyclin concentration or deactivate a CDK, and mark the cell for apoptosis or cell cycle arrest. Whereas, so you have cyclins all over these. So you have the G2 cyclins, you have the M phase cyclins, you have the G1 cyclins, right? Now, these are things like growth factors or nutrient availability. These can change how cyclins or CDKs work. So let's take a detailed case study of one of them. So this cyclin CDK can be phosphorylated at a place and three places actually. So the first phase is the one which activates it, and the two other places are where it's deactivated. So those two places, the phosphates need to be removed. So phosphatases uh, remove them, and then it's the active form. And now this will phosphorylate some other proteins. For example, laminin protein is phosphorylated and it destabilizes the nuclear membrane, right? And this can also activate another protein called anaphase promoting complex. Now, anaphase promoting complex is a ubiquitin ligase. That means it will add a protein to ubiquitin ligase add. Now, ubiquitin addition would mark it for, let's say, destruction. So this is just one example of how CDKs, once activated, they can change. So let's talk about a protein called cohesin. Cohesin binds the chromatids together, and separase, a protein, wants to break it. But there's a protein called securin that keeps on inhibiting this separase, right? And hence the cohesin, that the chromatids are apart. But once the anaphase promoting complex is active, it will it will break apart the securin. That means separase would be activated, and that means the cohesin would be degraded, right? And the chromatids would be split apart. So you see how a chain of events starts by just one phosphorylation and two dephosphorylations of the cyclin CDKs, right? So APC itself further degrades the cyclin, and hence the CDK is activated. So this is how the G2M transition is controlled, right? Now G1S or G1 cyclins, they are the different cyclins, and several things can activate or deactivate them. They are very different cycles, right? So for example, if we talk about G1 cyclins, so once it's activated, there's this E2F is a transcription factor. It would allow the transcription of genes for DNA replication. Now there's an RB protein that inhibits this E2F transcription factor. But once the CDK is active, this RB protein is inactivated. That means the E2F transcription factor is free to work. So you see how at various stages the cyclin's activity can really change this. Now growth factors, they activate CDKs. So different things, okay, by different pathways can activate or deactivate these things. Now next we'll talk about meiosis and recombination in detail, but before that we need to practice. So this is question number 1. I have 2 questions in this video. So this is question number 1. Try it yourself and leave the answer down in the comments, and we'll come up with a video to explain you this. This is the first question. There's one more question, and I want you to take screenshots because you need to practice. Once you practice, your some questions would be very easy. Like this one is perhaps an easier question, right? And there might be some questions which is difficult, right? The more you practice, the more you will get a hang of it. Now this is a difficult question. So here you might see different kinases, different things that can activate a cyclin. So read the paragraph and the diagram, and there are 4 questions. You have to just mark them true or false, right? But remember, you don't need to mug up any information for this. All that you need is given already in the diagram or the paragraph, right? So best of luck, and I'll make sure to bring a video to explain you this. And make sure you check out other videos as well, and do remember to visit our channel for the solution as well.
We hope this video was helpful. To see more biology topics covered, click on our channel and don't forget to subscribe.